welcome. Let me welcome you guys again. I'm fellow biochemist and of course biotex. So this particular video will be on model 2 of BCHEM 563 and BCHEM 565. Okay. So this particular model, we are going to talk about central dogma of molecular genetics. We'll be looking at transcription and translation. Okay. So I think I did mention of the central dogma of molecular genetics and what it's actually comprised of. Okay. So you realize, let's look at this little diagram over here. This is a DNA. Okay, the DNA actually unwinds into two single strands, and one of the strands is used as a template, and other one as like, no, let's forget about the other one. The other one, we are not going to talk about it, but the other one will be like the coding strand. Okay, but the one, the template strand is going to be used to transcribe an mRNA, and the mRNA is going to be transported out of the nucleus into the cytoplasm where translation occurs. Okay, and you see some structure here that's helping us, synthesize our polypeptide, and we're going to look at all of this. Okay. And open reading frames, there are some terms I think you should know. I'm not sure they are defined. So open reading frames, I think I'll need a diagram. Oh, I can't draw. Why? Who are these people? I wanted to draw. So you see, you have a gene. Let me see if there's any structure. Well, let me just narrate it. It's unfortunate I cannot draw. I don't know why, but so you can have a gene, right? You can have a gene, and there will be a start codon and a stop codon. Okay, so the segment between the start and stop codon is what we call the open reading frame. Okay, like we can define it in our own terms as that segment is open to be read or to be read for coding. Let's put it that way. Okay, so until we get to the stop codon, the polymerase runs along coding or transcribing the entire strand, right? So from the start codon to the stop codon, that's what we call the ORFs. Okay. What am I missing? I think we can move on. So this is the structure of DNA. Okay, I don't know if there are any texts. I'm not sure there are any texts. So let's look at the diagrams. So we all know the DNA to be a double helix, right? And it's comprised of 4 bases: A, T, G, and C. Okay, A and G, or adenine and guanine, are actually, so here are they. A and adenine and guanine are actually purines. Okay, and pyrimidines, cytosine and thymine are pyrimidines. Okay, so when we are going to talk about RNAs, it will be cytosine and uracil instead of cytosine and thymine. So thymine is going to be replaced in RNAs by uracil. Okay, so purines are double ring structures, and the pyrimidines are single ring structures. Difference between the guanine and adenine, what you have to know is purines are not methylated. Okay, they don't have a methyl group. The difference between them is, at this point, guanine has an amino group here and an O here. Okay, so you realize the position where there's an amino group on adenine, that same position on guanine is replaced with an O, and its amino group is found on a different place, on a different carbon. Okay, so adenine does not have an O group, but guanine does. Right. Cytosine is this is cytosine. Okay, this is cytosine, and thymine is just a deaminated but methylated cytosine. So when cytosine is deaminated, that means the NH2 is removed and replaced by a methyl group CH3. We get a thymine. Okay, but when cytosine is deaminated without any methylation, we get uracil. Okay, so please take note of these structures as well. So those are the 4 bases we use, and according to Watson and Crick, I'm sure that those are the names. Watson and Crick, Watson's principle or model or whatever, base pair rule. Yes, A binds to T and G binds to C. Okay. Looking at the conformations, you realize when G binds to C, they are forming 3 bonds, and when A binds to T, they are forming 2 bonds. So from our organic chemistry, we know three bonds are obviously will be stronger than two bonds. So we see that when you see a gene or a sequence of genes, let me put it that way, and it has more GCs than ATs, that particular part of the gene is more stable than another part of the gene that has more ATs than GCs. Okay, so if you are given a DNA sequence with more GC content than AT content, it is, I wouldn't say stronger, but more stable than another DNA with more ATs than GCs. Okay, because of the number of bonds. Okay, hence they will require higher temperatures for denaturation, those with higher GC content than those with higher AT content. Okay, so if you take the structure of a DNA, you have nucleotides. Nucleotides are the building blocks of DNA. Okay, a nucleotide is made of a sugar. Okay, in DNA, the sugar is deoxyribose sugar, right? I don't know if it shows here. Okay, here it is. So this is a deoxyribose sugar. You realize at the 2' carbon over here, it's actually supposed to be an OH, but because there is no O, it's actually deoxygenated, so we call it a deoxyribose sugar. Okay, it has the 5' carbon bound to a phosphate group. Okay, and the 1' carbon, or the position 1, bound to the base, either one of the 4 bases. Okay, so the base, sugar, and the phosphate group forms a nucleotide. When the base and the sugar comes together with no phosphate group, we call it a nucleoside, right? So that's how a nucleotide looks like, and multiple of these nucleotides come together to form our DNA strand. Okay, so this is one strand, this is another DNA strand. So you realize this is your sugar, this is your sugar, this is your base, and this is your phosphate group. Okay, so the phosphate group is going to bind to another phosphate group of a different it's going to bind to what is it going to bind to? So this is the phosphate group of this is a phosphate. So the phosphate group is going to bind to the next nucleotide. I don't know where. I actually do not know where, but it looks like at the OH, this OH, I'm sure that OH leaves it, makes it more reactive. Okay, so it's going to bind to that OH. So the phosphate of the first nucleotide is going to bind the OH of the second nucleotide, and that's how the nucleotide chain is formed. Okay, so the backbone, or what holds the nucleotides together in a strand, we call it a phosphodiester bond. So the bond that forms between the phosphate group of the first nucleotide and the OH of the next is called a phosphate diester bond. Okay, so this is how a DNA strand is formed. Okay, so when two complementary strands lie side by side, they are obviously going to be spare. Okay, so when A finds T, is going to be spare. G, C, ATs are right. I think we've talked about all this. So they actually, when a DNA twists around when a DNA forms a double helix structure, we actually have major and minor groups. Okay, and I recall in the major groups, that's where most DNA protein associations occur. So when a protein has to bind to a DNA for any sort of reaction, that they mostly occur in the major groove, okay, and not in the minor groove. And looking at how the spiral structure of the DNA is, the major grooves are more obvious or consistent than the minor grooves. Okay, I think you can even tell by just looking at it. And they also have, the bases are stacked on each other, and there's this pillar-like structure in the center of the double helix structure, and this also adds to the stability of the alpha helix of the DNA. Okay, we also have 3 different types of DNA. We have the B form, A form, and the Z form. Okay, so we are going to take them one by one. The B form is the most common, or yes, let me call it the basic form of DNA. So almost all normal DNAs are of the form B, right? And we call it a left-handed DNA. That's a helical sense. Okay, so it is actually twisted in the right-handed direction. Okay, and it has a major group present. So if you turn, so this is, let me tell you what a helical turn is. So when it moves this way, you see it is moving like a spiral manner. So like this one circle is a helical turn. Before it turns another one, second helical turn, third one, fourth one. So per helical turn, for the B DNA, it has 10.5 base pairs. Okay, so we are going to find 10.10 and a half base pairs per each helical turn. Okay, and it has anti-glycosyl bond formation, and the helix rise per base pair, it's like the diameter, or the um how wide each turn is. Okay, this is 3.4 Armstrong. I don't know if that's Armstrong, but I'm saying it's Armstrong. Okay, so in B DNA, the how wide the helical turn is 3.4. Okay, and the base tilt is 6 degrees. I'm not sure this is important, but make sure you know this. So this is the structure of a B form DNA, and the A form is a bit similar, also right-handed, has major groove, but it has 11. This is wider than the A form is wider than the B form. Okay, no, sorry, it is narrower than the A form. Okay, so it is more stretched, so it has 11 base pairs per helical turn. So you realize it has um a narrower diameter. That's 2.6 Armstrong. Okay, and the one that differs the most is the Z form, which is left-handed. Okay, mostly these are interchangeable. Okay, depending on conditions in the organism. So B can convert to Z, and when conditions are favorable, or I think there's a particular process in sporulation. Okay, and when that is supposed to happen, you realize the B form DNA will actually convert to Z, and some things will happen, right? So when conditions return back to normal, the Z form DNAs are going to reverse to the B form. Okay, these are not like aberrant DNA shapes or something, right? So these Z forms are left-handed. They do not have major grooves, and they are a bit wider than the previous two we've talked about. They have sorry, yes, it's actually wider and longer. Okay, so it has 12 base pairs per 10. Okay, and it is actually wider with 3.7 Armstrong diameter. Okay, and this is how it looks like. Not exactly, but this is actually supercoiling, and there's no much to show. Much to show. Okay, supercoiling is just after the DNA forms a helical structure, it actually undergoes supercoiling, and I'm sure this will be talked about when you are talking about how DNAs are folded, okay, into chromosomes, the chromatins to fit into the nucleus, blah blah blah. Okay, so we are just twisting the helical structure or the helix around. Okay, and these are actually facilitated by the enzymes we call topoisomerase. Topoisomerase can undo or do supercoiling. Okay, so I think I'll just skip this, and we are going to discuss it when we are doing chromosome or chromatid folding, right? So let's look at DNA replication and what it is. DNA replication simply means, DNA is made up of two strands. So the strands separating into single strands, and free nucleotides or free complementary nucleotides are going to bind to each strand, and we are going to get two different daughter cells from one DNA molecule, right? So a single strand starts replicating. At the end, you are going to get two daughter cells. Okay, so DNA has a polarity, and the two strands has to be anti-parallel. They shouldn't be parallel. So the 5' end should be the same direction as the 3' end of the other strand, and not the other 5' end of the other strand. Okay. So yes, when we are adding nucleotides, when you are doing polymerization, okay, when you are growing a chain, we add new nucleotides to the 3' end of the strand, okay, or the new molecule, and not the 5' end, okay. So it's mostly the OH that determines the reactivity of the molecule, okay. That's why the new nucleotides are added onto the 3OH end of the chain. So this is an example of a DNA structure unwinding into two strands, and those strands being synthesized into new molecules, okay. And there were these, should I call them, theories about how the DNA replicates, okay. There were actually hypotheses. Some, what we know of the DNA replication is the DNA replication is actually semi-conservative, okay. And there are other propositions as well. Some said it's conservative, some was like is it dispersive, okay. Some were like it is dispersive. So the semi-conservative is what we know. When a DNA splits up into two, each strand from the parent molecule gets three nucleotides that are complementary to the strand, of course, and forms two daughter cells. And those two daughter cells each have a single strand identical to the parent molecule. That would be like the blue, and the yellow is the new one, okay. But for conservative replication theory, it says that one strand will be entirely similar to the parent molecule, and another different from the parent molecule. And the dispersive is like different part of, so you can't, the dispersive is like no one strand is exactly as the parent molecule, but there are segments of each strand that are actually from the parent, okay. So like piece, not like a full strand from a parent molecule, right? So these are some reactions done, or experiment done, okay, to predict whether we are doing a semi-conservative replication or conservative or dispersive, right? So for the semi-conservative, you realize the DNA in the nitrogen 15 way is heavy, okay. So it's going to be at the bottom, right? But that's the parent molecule. But when they divide, and now, so the parent molecule is actually grown in, or is labeled with the blue structure, the blue color, that's N15, and the new nucleotides are labeled with N14, okay. And the N14s are actually less denser or less heavier than the N15 ones. So the pure structure, which is the parent molecule, are going to be heavy, and of course they will be at the bottom of your test tube. And when it grows, and truly they are it's actually a semi-conservative replicative model, you are going to get a hybrid, one N15, one N14, okay. And that will be like in the middle, okay. It won't be at the bottom, okay. So let's just focus on the first generation, because the first generation is where you can determine that yes, it is a semi-conservative or not a semi-conservative replication. They did the same thing to predict whether it is conservative or not. So let's focus on the first generation. So of course the parent molecule will be down here, but this time around they got just the two strands they got, or the two daughter cells, daughter molecules they got, one was down and one was on the upper side, showing that one was entirely similar to the parent molecule, another entirely different, okay. And this is how we predict the conservative. Dispersive is something like this, okay. So dispersive too is going to act like the semi-conservative, and I think this is where the second generation plays a role, okay. So you do second generation replication, you realize it will be floating around in the middle, but not exactly where you found the first generation molecules, okay. So let's talk about some enzymes or proteins, okay, that we employ in DNA replication. So we are going to look at these enzymes in prokaryotes and eukaryotes, okay. So first we are going to look at the enzymes in prokaryotes. An example is in E. coli. So these enzymes are going to look at our DNA polymerases, okay, and there are classes of them. There are 5 types of them: polymerase 1, polymerase 2, polymerase 3, 4, and 5, okay. So the genes that encodes for these enzymes are called pool A. Pool A codes for polymerase 1, pool B codes for polymerase 2, pool C polymerase 3, din B polymerase 4, umu D2C. So these are the functions. Okay, the last two, 4 and 5, are less talked about in most cases, especially in our course. Okay, and what we know about them is they are used in translation replication, and we are going to talk about it as well, not into details, but you should know that translation are like some, like, call them mistakes. Okay, so when there are some mistakes or mutations in the DNA, a normal polymerase cannot synthesize or cannot synthesize that particular strand. Okay, so these polymerases, the polymerase 4 and the polymerase 5, are recruited to help synthesize past those translations. Okay, then the normal polymerase will come in to continue the replication. So polymerases are just, when you hear polymerase, polymerase means an enzyme. ACE is an enzyme, right? So a polymerase simply means an enzyme that's used to polymerize or synthesize a new strand. Okay, or let me not say new, synthesize a strand of a molecule. And in this case, our molecule is a DNA or nucleic material, be it RNA or DNA. Okay, so pool 1, polymerase 1, is actually like, and not entirely but majorly, a repair enzyme. Okay, we are going to look at them individually as well. Pool 2 is for replication restart, and pool 3 is mainly a replicase. Okay, so it plays a role mainly in replication, and pool 1 mainly as a repair enzyme. Okay, so this is the replication, what's the name given to it? Is it replication, replisome or something, of a prokaryotes? Okay, so this consists of a DNA unwinding and replication of the two daughter strands occurring in the different factors or proteins that are recruited or that take part in prokaryotic DNA replication. Okay, so what we can see, let's see what we can identify over here. We can see topoisomerase. Okay, and in this case, this is preventing supercoiling. Okay, and we can see a helicase. This helicase is what is unwinding the DNA strands into single strands. Okay, so the function of a helicase is to unwind double-stranded molecule into single strands. Okay, so when the helicase unwinds the DNA molecule, we have these small proteins. What are they called? Single strand binding proteins. Okay, these are making sure that no random nucleotides bind to the strand. Okay, or the strands do not get back together, right? So these single strand binding proteins are making sure, or are preserving, the strands for the synthesis of a new daughter molecule. Okay, and we can see a primase, right? So what, I'm not sure there's a diagram explaining this. We'll come here. So we have the two strands over here. They are different. Synthesis of the two strands are different. Okay, so we have something called the replication fork. Okay, the replication fork is where the unwinding, the direction where the unwinding is going. Okay, so the two strands that are separated are replicated or are synthesized in different directions. The one going in the direction of the replication fork is called the leading strand. Okay, and the one moving away or in the opposite direction of the replication fork is called the lagging strand. Okay, not that the leading means is going to be synthesized or replicated first, or the lagging is going to be replicated or synthesized last. No, the one in the same direction as the replication fork is going to be, it's called the leading, and the one in the opposite direction is called the lagging. So in the leading strand, synthesis occurs continuously. Okay, so you realize there's not much occurring on this strand. So what happens is this DNA polymerase, the DNA polymerase 3, okay, is actually responsible for the synthesis. So if you go back to this table, you can see that DNA polymerase 3 is the actual replicase. Okay, so it's the main polymerase enzyme responsible for replication or DNA synthesis. Okay, so DNA polymerase 3 is a dimer, one subunit on the first strand, the other subunit on the second strand. Okay, and there's a clamp. The clamp is called, in prokaryotes, it's called beta clamp. It will have a different name in eukaryotes, and you go there. The clamp holds the DNA polymerase 3 intact, okay, preventing it from falling off. Okay, and this is actually it plays a big part, the clamp, differentiating it between the polymerase 3 and other polymerases, because other polymerases are not constitutive, they cannot stay on the strand, that's why we don't use them. Okay, so this is what differentiates the polymerase 3 from other polymerase like polymerase 1, even though polymerase 2, sorry. So polymerase 2 is also a replication polymerase, but because it cannot stay on the strand for a longer period of time, we don't use that. Okay, so the beta clamp holds the polymerase in place, and the leading strand will be synthesized from start to finish, or start to end, right? Sorry, but that's not the case for the lagging strand. For the lagging strand, for whatever the case is, they are synthesized in pieces. Okay, and actually, because they are synthesized, so let me take one step by step. They are synthesized in fragments, and I don't know if I should attribute this to the fact that they are moving away from the replication fork. Okay, so they are not synthesized continuously like the leading strand is synthesized. Okay, so you need a primer. Okay, of course you are going to need a primer. Is just prime means, I don't know if anyone understands Spanish, but primer is actually gotten from the word first. So something that initiates something. So a primer is just a sequence that is going to initiate the synthesis reaction. Okay, so the protein is the primase or RNA primase. It's going to synthesize for the primer. Okay, the primer will be what is going to initiate the reaction. Then DNA polymerase 1 will start. So the reason why we are needing a primer is because the DNA polymerase 3 does not have a primase action. Okay, it cannot start replication, it can only continue replication. So the primer has to start it. Okay, the primer starts on the leading and the lagging strand. Okay, but on the leading strand, you only need one primer. But on the lagging strand, because we are synthesizing in fragments, after the end of each fragment, you need a primer to start the next fragment. That's why you are seeing so many reds. One here, one here, one here, one here. So, right? But I'm not seeing any red here. Okay. So, the fragments that we synthesize on the lagging strand are called Okazaki fragments. Okay. So, the first fragment is called Okazaki. Second one, Rasa. So, after the synthesis of the Okazaki fragments, the primers are deleted. Okay. Then we join the small fragments. Okay, and this will form a full daughter molecule. Okay, and I think after deleting, this will be the latter part of the slide, but let me just mention it. After deleting the primers, you realize this lagging strand will be shorter than this, and it is called end is it end replication problem or something? Yes, and we talk about the latter part of this slide. Okay. I think these are all that we need to know. And there's a ligase as well. Ligase are what, after the removal of the primers, ligase is the enzyme that joins the fragments. Okay, and we can see it from here. So this is the synthesis on the lagging strand. Okay, so you realize these are the primers, the red ones. So it will be synthesized in fragments. So in this case, we are showing two fragments. This primer initiated the synthesis of this fragment, and this primer initiated the synthesis of this fragment, right? So after assuming this is the whole length of the strand, after synthesis, the primers will be degraded. Okay, so the primer comes in. Okay, the primase synthesizes and synthesizes the RNA primer. Okay, so the RNA primer is synthesized by a protein called primase or an enzyme called primase. Okay, and after the primer initiates the reaction, DNA polymerase 3 comes in. Okay, to elongate or synthesize the strand. And after everything, DNA polymerase 1 comes in. And I think you know what DNA polymerase 1 is from the table. DNA polymerase 1 is a repair enzyme. So it comes in to clean mistakes. Okay. So DNA polymerase 1 comes to degrade the primers, okay, from the strands. And the ligase will come in to join the Okazaki fragments. And this is the ligase over here. Okay. And that's how the lagging strand is synthesized. But it wasn't that complex in the leading strand. The leading strand is synthesized continuously. Okay. So that's for prokaryotic DNA replication. It's similar in eukaryotic, or should I say it's the same in eukaryotic replication, DNA replication, but the factors of the proteins are different. Okay, even though most of them perform the same function, the names are different, so you have to take good notice of this. So the name of the polymerases are in, I don't know if these are Latin or Greek alphabets. DNA alpha, is this delta, epsilon, gamma, beta, blah 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 blah, right? So with the polymerases, if you see a polymerase and a number comes after it, or a Roman numeral comes after it, that's a prokaryotic polymerase. And if it is a polymerase, I'm calling this Latin alphabet. Okay, so if it is a, if the polymerase is with a Latin or Greek alphabet, the alpha beta stuff, that's a eukaryotic DNA polymerase. Okay, so with the DNA polymerases, this table, I think we should consider it very important. Very important. I don't know. For Doc, everything is possible, right? I don't expect him to ask us, what's the function of polymerase A, alpha, or this, but I think we should know. Okay, so the polymerase alpha, which we can liken to the polymerase 1 in prokaryotes, is for nuclear replication. Okay, so the polymerase alpha, polymerase delta, epsilon, and gamma are replicase polymerases. So they are actually involving replication. Okay, and beta, the ZNIK, those are repair enzymes. Okay, but we have high fidelity and low fidelity polymerases, and mostly the repair polymerases are low fidelity, except the polymerase beta. Okay, so the unlike the prokaryotes, you see in prokaryotes the same polymerase 3 synthesizes leading strand and lagging strand, but in eukaryotes the lagging strand is synthesized by DNA polymerase delta, or polymerase delta, and the leading strand is synthesized by polymerase epsilon. Okay, we have a look. So realize in prokaryotes the same DNA polymerase 3 was synthesizing the leading strand and the lagging strand, but it's not so in eukaryotes. Eukaryote, the leading strand is synthesized by polymerase epsilon, lagging strand is synthesized by polymerase delta, different polymerases. Okay, and just like the prokaryotes, they have a clamp, but this clamp is not called beta clamp, it is called a PCNA. Okay, and it also has the eukaryotes also have a helicase. This helicase is called, we can in eukaryotes we call it MCM complex. Okay, I think almost everything else is similar, but one, I don't know if it's stated. Okay, it's not stated, so I should talk about it. Another thing is the polymerase alpha. Okay, this polymerase is what primes the reactions. So the polymerase alpha, even though it's a polymerase, it has a primase activity. So in this case, you are not going to need a primase to synthesize your RNA primer for you. So you are recruiting polymerase alpha, which has a primase activity, to prime our reaction for us. Okay, so after primase, it falls off, then the polymerase delta and polymerase epsilon respectively synthesize their strands. Okay, let me read this. Eukaryotic cells have many DNA polymerases, blah 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 blah. The three different DNA polymerases make up, I think I've said everything over here. Yes, I've said everything. So one term you can give to the work that the clamp does is it makes, or it endows the polymerase with processivity. Okay, so the fact that it makes it stay on the strand without falling off, we call it processivity. Okay, processivity. Yes, so this is a comparison between the factors or the proteins in prokaryotes and eukaryotes. So you can realize DNA B, this wasn't stated. So the helicase in prokaryotes is called DNA B. Okay, and in eukaryotes is the MCM complex. Right, this is in viruses, but you're not focusing on viruses now. CDC6, this wasn't also showed, but I think CDC6 and CDT blah blah are here, right? We've not actually talked about it, but its homologue in prokaryotes is called the DNA-C, and they are needed to load the helicase or the primase on the strand. Okay, so you need a protein to recruit the helicase on the, so before the so the replisome is not like something like constant. So I think in third year in the slides there's a particular page that talks about the assembly of the replisome. Okay, so before the replisome or all the proteins come together, something happens. Okay, different factors bind to things, causing some things to do. Okay, and some of the things that happened was DNA-C. Okay, so the first thing was DNA-A binding to blah blah blah, I've forgotten, but DNA-C will bind to something to recruit the helicase. Okay, so until then, the DNA strand cannot be unwinded or unwound. The DNA strand cannot be unwound or splitted in two. Let me use that. That's much simpler. So the DNA-C is responsible for recruiting helicase or loading the helicase, and RPA. This is the maintenance. So the little proteins on the single strands. Okay, in prokaryotes they are the single strand binding proteins. In eukaryotes they are called RPAs. The primers in prokaryotes they are the DNA genes. Okay, that's like the primase or the RNA primer. In eukaryotes is the polymerase alpha. Because it has a priming activity, we call it polymerase alpha primase. Okay, I think you can let me skip this and have time with this and play with it as well. So let's look at DNA replication and the yeast cell cycle. Okay, DNA takes place in the S phase. So during the cell cycle, I think we have different stages. We have J1, J2, and S. So the S phase is when DNA synthesis takes place or DNA replication takes place. Okay, so in yeast, the method of control of the S phase is linked to replisome assembly of the blah 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 blah. So I think there's a diagram. Okay, there's a diagram, not this. Okay, there's a diagram here. We will come back to it. So in yeast, there are 3 proteins that are required to begin the assembly of the replisome. Okay, as I said before, all the proteins come together on the DNA. Okay, so when all the proteins are recruited, the polymerase, the topoisomerases, helicase, ligase, polymerase 3, like all of them, when they are recruited on the DNA and replication starts, the whole thing is called the replisome. But before all of them are assembled, 3 proteins in yeast, 3 proteins are required. Okay, and the first one is the ORC, which is the origin recognition complex. Okay, so this ORC is the first thing. It binds first to a sequence in yeast origins. Okay, so yeast also have their origin of replication, and the ORC will bind to the origin of replication. Okay, so in E. coli, as I said, it was a DNA binding to the ORC or the origin of replication, and in E. coli, when the DNA binds, then the DNA-C will recruit the helicase. Okay, so if you look here, DNA-C was for E. coli, and CD6 is the homologue in eukaryotes. Okay, so now we are talking about eukaryotes, which is a yeast. So we are not looking at DNA. Okay, so first the ORC binds to the yeast origins. Okay, so when they bind, then they are going to recruit the CDC6 and CDT1. Okay, CDC6, CDT1, and the ORC all being in place are going to recruit or call for the MCM complex, which is the helicase and other components of the replisome. And when this happens, you know what the helicase does? It starts the unwinding. Okay, so this is it. This is the origin of replication. Realize the ORC comes in and binds the origin, and after binding, the CDC6 and the CDT1 are going to come in. When they come in, they recruit the helicase and other polymerases. So the helicase will start unwinding, the polymerase will come in and start synthesizing. Okay, so before this coming, the replisome is not assembled, but here you realize the replisome is now assembled. Okay, So just take notes. In yeast, before assembling of the ribosome takes place, there are three proteins that are needed to, recruit all the proteins needed. So this is the S phase in the cell cycle where replication begins. Okay, so replication is linked to the cell cycle through the availability of CDC6 and CDT1. So what actually happens is after replication starts, the CDC6 and CDT1 are destroyed. Okay, so that replication can go on like successfully, right? These proteins are synthesized during late mitosis and J1 phases and are destroyed by proteolysis after synthesis has begun, as I said. In this way, we make sure that replication can be assembled only before the S phase, okay? And when replication begins, new replications cannot form. So each cell, let me say, replication happens once at a time and not multiple simultaneously, okay? So when one replication begins, the CDC6 and CDT1 are going to be destroyed so that one particular replication can go to completion, okay? Now let's talk about telomeres and telomerases. So telomerases are the enzymes or the proteins that code for this telomeres, okay? And telomeres are some end structures. So they are like sequences, but these are not coding sequences, okay? And they are mostly found at the tips or the ends, the terminals of DNA molecules, okay? So bidirectional replication process in eukaryotes comes with a major problem. I wish I could draw. I don't know why these things are not available. Sorry. So I'm sure we discussed this in class, but unlike the prokaryotic chromosome, eukaryotes, we have multiple origin of replication. Okay, so replication is bidirectional. Replication moves to the right, to the left, like in different both to the left and to the right. But in eukaryotes, because they have only one origin of replication, it is a unidirectional replication. Okay, and in eukaryotes, because ours is bidirectional, it causes like major problems, right? And it can replicate most chromosomal DNA, but there is an inherent problem in replicating telomeres. So let's go what telomere is. Telomeres are specialized structures at the end of chromosomes, as I said, and they are added or they are encoded for by the enzyme called telomerases. Okay, so telomeres are mostly or actually added to the 3' ends of your DNA structures. Okay. I think I've mentioned the end replication problem as well. You realize when we are synthesizing the ligand strand, we have primers within it, and afterwards we have to remove the primers. And if you recall, we use DNA polymerase 1 to remove the primers, and a ligase will come and join the Okazaki fragment. So when this is done, you realize the ligand strand is going to be shorter than the leading strand, and we term this the end replication problem. Okay. So this is just a representation of everything I just said. The leading strand will go on to completion with no complications, but the lagging strand will end up being a bit shorter. But if this goes on, so after each round of replication, you realize the lagging strand will be shorter and shorter and shorter. Okay, so there should be a way of limiting this. I think this can be stopped, but there should be a way of limiting this. So cells have evolved a specialized system to combat the end replication problem. Okay, so a simple telomere, which is encoded for by telomerases, will be added to chromosome tips. Okay, so we add the telomeres to help us cope with this end replication problem, not to do away with it. We can't do away with it. Okay, so the telomeric sequences or the telomeres which would have otherwise been lost are added back to chromosome ends. Okay, and this helps the cell or the DNA not to lose like important genetic information. Okay, so the telomerase, the protein that codes for the telomere, okay, that's the telomerase. The telomerase is a ribonucleotide the telomerase is a ribonucleoprotein enzyme with two subunits. Okay, and these subunits have they all have each one of them has their own distinct functions. Okay, the two subunits are the tech and the tet. The tech is the telomerase RNA component. Okay, telomerase RNA component and telomerase reverse transcriptase. So let's talk about the RNA component first. The RNA component, of course, this is the RNA component of the telomerase. Okay, so what it does is it is an RNA subunit that's going to serve as a template for the synthesis of telomeric repeat unit. Okay, so the RNA component simply serves as a template for the synthesis of your telomere. Okay, because for you to synthesize something that's going to bind to something, you need a template. Okay, so within the protein is the tech, and that tech serves as a template for the synthesis of your telomere, right? The second subunit, the third, okay, is a protein subunit, is a polymerase. Okay, let me call it transcriptase, but it's a polymerase. So if you hear reverse transcriptase, you see mostly we synthesize RNA from DNA. Okay, but in this case, you are going to synthesize DNA from RNA. So any enzyme catalyzing these kind of reactions are called reverse transcriptase. Okay, so TET, which is a telomerase reverse transcriptase, is a protein subunit that harbors the polymerase function. Okay, so realize you are going to synthesize a DNA from RNA. Okay, so that's what it does. So it is going to use the RNA template to synthesize a DNA sequence, and that DNA sequence is what we are going to call the telomere. Okay, so synthesizing DNA from RNA is called reverse transcription. Okay, because the normal transcription, the normal sequence of things is transcribing RNA from DNA and not DNA from RNA. Okay. So this lady and this man, Elizabeth Blackburn and Joe Gao, what they did or what they said was ends of chromosomes are made up of repeated tandem arrays of these sequences in tetrahymena. Okay, but in humans, so in tetrahymena is TT but in humans it is TTAJJ. Okay, so these are like repetitive sequences that are going to find in all chromosomal ends. Okay, so you realize all eukaryotes have short tandem repeat, and this is so however they are not exactly the same, but when you see them you will know that okay, this is what this is the end of a chromosome. Okay, so I think there's a table here and it shows like the different end repeats in different organisms. I think humans, where are we? Vertebrates, the last part. TTAJJ. Whom are we similar to? Neurospora crassa. You realize they also have the same tandem repeat. Okay. So telomerase, after synthesizing the telomere, adds these short repeats to the 3' ends. Okay, and I think the telomere. So in humans, for example, the telomere is made up of a repeat of TTAJJ. So the telomere will be like TTAJJ, 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 like numerous of them. Okay, so the telomerase will add the repeat to the 3' end of the DNA. Okay. So of course, if the human chromosomes have a repeat of TTAJJ, the telomere will be what? The RNA sequence or the template is going to be complementary to the TTAJJ. So in the tech component, which is the RNA component of the telomerase, you are expecting the template to be AAUCC. So AA are complementary to the TT, U complementary to A, okay, and CCC complementary to the JJJ, right? So that's the RNA sequence in the tech component, serving as a template for the TTAJJ. Don't be confused. This is the RNA component. That's why I'm seeing U. But this tandem repeat of the telomere is a DNA, even though it was synthesized from an RNA. That's why it is actually synthesized by a reverse transcriptase. Okay, so it is a DNA segment. That's why it has a T, but the template has an U or a U, whatever you call it. So what does the tech does? The tech component with telomerase, it first anneals to the 3' end overhang. Okay, and it's going to extend it okay, using the telomerase's two components, using the tech as the template and the TET as the polymerase or the synthesizer, okay, or the transcriptase. So after adding few nucleotides to the 3' end, so this is like how it helps combat the end replication problem. So it binds to the where the shorter strand, that's the lagging strand, okay, the tech will anneal to it and synthesize or extend the overhang, okay. It moves along the DNA so that the 3' end can be further extended. There's a diagram here, we come to it. So it extends it, and the 3' end continues to be extended by repeated movement, blah 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 blah. Primase and DNA polymerase then use a very long as a template to fill in the end of the other DNA strand. So this is it. You realize the lagging strand is shorter than the leading strand, okay. So you bring in your telomerase. Your telomerase will come in and it will extend the leading strand, okay. So it is going to use a tech component as a template and extend the leading strand, right? So after extending the leading strand, then the primase will come in to initiate the synthesis of the lagging strand. Then the DNA polymerase part of the telomerase, which is found in the TET, okay, the reverse transcriptase part, will fill in the missing part. And mind you, after synthesis, you have to remove the primer part, okay. And even though here you still have a shorter lagging strand than the leading strand, initially where the information was missing is still filled, okay. So even though the end replication problem is not solved, you are not going to lose the information you're supposed to lose, okay. So initially you were missing like this part ACC, this is it, okay, from the GTT parts. So you're missing all these parts, okay, but now you are missing only this. So it will take a longer time or numerous cycles of replication to lose the information you would have lost if the telomeres weren't available, okay. So even though the telomeres help with the end replication problem, but they can't do away with it completely. I think I've said this. So there are two critical functions of the telomeres. One, the telomeres prevent erosion of genetic material after each round of replication, okay. And they also preserve chromosomal integrity by associating proteins to form protective caps. Okay, so there's this, these are some telomeric proteins, okay, and they called shelterings, okay. The sheltering form a cap at the chromosomal ends, okay. So that's another function of telomeres. They interact with proteins to form a cap, and these caps protect or preserve the integrity of chromosomes, okay. So double strand breaks in chromosome in DNA are potentially very dangerous, and they can result in chromosomal instability that can lead to blah 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 blah. So the cap that I said a telomeres can interact or associate with different proteins to form, this cap protects the telomere at the end of a chromosome, okay. It actually hides, in quotes, it hides the 3' overhang when it displaces the DNA strand and blah 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 blah. So these are part of the proteins that form the protective cap, okay. And with other proteins, they are called the sheltering, the TRF1, TRF2, RAP1, TIN2, TPP, and POT1, okay. I think there are six of them. Together they form a protein complex called sheltering, okay. The sheltering protect the telomeres from DNA damage, okay. So these are like caps, you can call it like helmets for the chromosome, okay. So as this particular this statement, yes, it was like double strand breaks are potentially very dangerous. They can lead to problems like cancer and age-related diseases, okay. So when double strand breaks are detected, the cell responds very quickly, okay, and it actually goes in two directions depending in part on the type of cell it is, or the type of cell that double strand break is found in, and also the extent of the damage, okay. So based on the type of cell and the extent of the damage, the double strand can be repaired, can be fused to another brick. Okay, let's say it can be repaired. Or if the cells can limit the damage, what they can do is they will stop further cell division. So that cell will be like, if it was a human, you call it infertile. Or yes, for guys they have another name for that, I've forgotten. So it will stop further cell division, and that cell will never get to replicate again, and that's senescence. Or when the damage done is irreversible or beyond tolerance levels, it will initiate cell death pathways called apoptosis. Okay. So let's look at some disorders or disease associated with this telomeric or shortening stuff. Okay, there are cancers and aging as well. So this in aging, it is mostly in the somatic cells. Okay, so the reason why telomeres are very important in germ cells or um, these your chromosome 23, these cells, right? The reason why they are very important is these cells contain the genetic information that has to be passed on to your offspring, so they have to be protected. But that's not the case in somatic cells. Okay, so somatic cells, they produce very little or even no telomeres. Okay, so as your cells continue to replicate over time, your DNA length is going to reduce. Okay, and that's what brings about aging. Okay, but there are some syndromes or there are some disorders. Sometimes there will be some humans who have shorter than normal telomeres. Okay, and this will make them look older than their age. And such an example of such disease is the Werner syndrome. Okay, this the early onset of many age-related events, including wrinkling of skin, um, contraction of cataract, osteoporosis, graying of hair, and cardiovascular diseases. So what actually happens is there's a mutation in a gene called the WRN. I don't know if it's a Werner gene or something. Okay, and this WRN is a gene responsible to code for a protein that's going to associate with proteins that produce the protective cap. Okay, so it is a gene that codes for one of the proteins in sheltering. Okay, we know the sheltering forms the cap, the protective cap at chromosomal ends, protecting the telomeres, right? But this particular gene, the WRN, is mutated. So in this case, the cap is either non-functional or even non-existent, right? And in this case, chromosomal ends are not protected, meaning telomeres can easily be shortened. Okay, so with shorter, with few cycles of DNA replication, they are going to be, they are going to look older than they actually are, right? And this, we call this premature aging. Okay, so this is someone who was 15 years, and after a few years, you realize after some few cells replication cycles, look at how this is happening. So another disorder is called the sclerosis congenita. Okay, there's another premature aging syndrome, and the phenotypes of this disorder is nail dystrophy. I think you can see that from here. Changes in skin pigmentation, mostly around the neck area. Okay, and especially and some white patches inside the mouth, and we call this oral leukoplakia, and also bone marrow failure, leukemia, and also some other cancers as well. Okay, patients have shorter telomeres, of course, than healthy people, and there are obviously mutations in some genes that are associated with telomerase activity. Example of these genes are the TEC in telomerase, DKC1, and TIN-F2. TIN-F2 also is in the sheltering proteins, right? Tin. This is TIN-2. Okay. So just like the telomere length is associated with aging, telomere length is also associated with cancer. Okay, So cancer, what they've done is one from hydrogen, unlike the normal somatic cells, which have like no little to no telomeres. These cancer cells have their own telomeres, so they are more of like immortal cells. They never get shorter or they never grow old because their telomeres prevent the degradation, or it makes them stable. It makes their cells stable. Okay, so their ends are not lost or are never degraded. Okay, so because the cancer cells have telomeres and normal somatic cells do not, these cells are always proliferating, they are always replicating, right, without losing part of their cells. And that's how the onset of cancer comes about. Of course, there are different factors causing each one of these different cancers we have, right? So this particular model will be in two parts. So this at part one ends at page 37. I'll see you in the next one.